Hello everyone, welcome to today's session. Today we are going to see Class 12 Biology, Chapter 5, Molecular Basis of Inheritance. In that we are going to see genetic code. So in the previous sections we studied that DNA is a genetic material, right? And that genetic information is transferred from DNA to RNA and finally it is used for protein synthesis. So what happens during replication? See, the DNA produces another DNA molecule, but during transcription, what happens? The DNA information is copied to RNA, and these processes they can easily understood because complementarity base pairing occurs between the nucleotides. So in DNA, when we take it is ATGC, right? But what happens in during the transcription? A pairs with U and G pairs with C. So this translation is nothing but the situation here is different, right? And also the information must be transferred from a polymer of nucleotides to a polymer of amino acid, and there is no direct complementarity between nucleotides and amino acids. So they require a special system in order to connect the nucleotide sequence with the amino acid sequence, and this system is called as the genetic code. Let's start with the topic. So, genetic code. So, there is no complementarity between the nucleotides and amino acids, and they need a code in order to link them. And now, why a triplet code? Yes. See, Gamo's idea is Gamo's idea. See, now let us understand why this genetic code is made up of three nucleotides. We already know there are only four bases in RNA. One is the adenine, yes, uracil, guanine, and cytosine. But these four bases have to code for 20 different amino acids. How many amino acids? 20 different amino acids. And George Gamow. So George Gamow so he proposed that the four bases must combine together to form a code. If one base formed one code, then it will be how many? Four combinations, right? And this is not enough to code for 20 amino acids. If two bases are coding means what will happen? It will be like a 4 square, right? So it will be 16 combinations. Again, this is not enough. But if three bases are combining, what will happen? 4 power 3. Yeah, it will be 64 combinations. So now we have 64 combinations you can see here. So which is more than enough to code for 20 amino acids. So Gamow he proposed that the genetic code should be a triplet code. He proposed that genetic code should be a triplet code. A group of three nucleotides is called as a codon. How many? A group of three nucleotides is called as a codon. For example, AUG is our codon, right? UUU and then GCU, right? So next we are going to see scientists who are involved in deciphering the code. You can able to see here S code deciphered by so many scientists name I have given here. So what they did, they proposed that triplet code was only the beginning. Yes, and scientists had to experimentally determine which codon corresponds to which amino acids. So several scientists have contributed to deciphering the genetic code. First one is George Gamow. So what he did, so George Gamow again he proposed the idea that a genetic code should be made up of three nucleotides. Just now we saw. And the next one is Hargobind Khorana. So what he developed chemical methods in order to synthesize the RNA molecules containing that defined combination of bases. So he synthesized homopolymers and copolymers. So what are they? So these are like RNA molecules. Yes, they help the scientists to understand the relationship between specific nucleotide sequences and amino acids. And next one scientist name is Marshall Nirenberg. So he developed a cell-free system for protein synthesis. What he developed? A cell-free system for protein synthesis. So what does system do? See, this system, it allowed the scientists to study protein synthesis outside a cell-free means outside the living cell, and also to determine how particular RNA sequences directed the incorporation of specific amino acids. So next scientist is Severo Ochoa. Yes. So this enzyme polynucleotide phosphorylase, yes, is associated with the Severo Ocho's work. So what he did, he helped in the enzymatic synthesis of RNA and with the defined sequences in a template independent manner. So what George Gamow did, triplet code he proposed, and also Hargobind the Corona, he defined the RNA molecules, and Marshall Nirenberg, he did the cell-free protein synthesis, and Severo Ochoa, he did the RNA synthesis. Okay, all these genetic codes are deciphered. So remember all these things. These things are very important. Let's move on to the next one. So here we are going to see the codon table and its salient features. So this is a very important thing. Okay, so these are the important properties of genetic code. So scientists, they prepared a codon table, and also they called this as a genetic code checkerboard. It's looking like a checkerboard, right? So this is genetic code checkerboard. So this table tells us which codon codes for which amino acids. You can see so many codons over here, and also the amino acids also given over here. So which codon codes for which amino acids? For example, let us take a where is this AUG? So this is a methionine, okay, and then UUU is a phenylalanine, and another phenylalanine. This is also UUC is another phenylalanine. So let us understand the important properties of the genetic code. Here I have given some important properties. The first one is the triplet. So triplet is nothing but the genetic code. You can able to see now this genetic code. So is a triplet code because each codon contains 3 nucleotides. You can able to see, right? So each codons contain 3 nucleotides, and there are how many 64 possible codon. How many possible codons are there? 64 possible codons are there, and then among them 61 codons are they code for amino acids, and 3 they do not code for amino acids, so they are the stop codons. Yes, the 3 codons, they do not code for amino acids. Yes, they are the stop codons. So what are the stop codons? So here you can able to see UAA, UAG, and also UGA. These are stop codons. They do not code for any amino acids. And next one is the degenerate. So the genetic code is called degenerate because some amino acids are coded by more than one codon. For example, let us take this phenylalanine. So where and all you can able to see this. See, this is phenylalanine and this is also phenylalanine, right? Yeah. And so what happens? See, this means that different codons can specify the same amino acid. And next one is the contiguous. So the genetic code is read in a continuous manner. How they are read? They are read in a continuous manner. So there is no punctuation marks or gaps between codons. For example, if you are going to read, how we can read? We can read as a UUU, UUC, UUA, UUG, right? So no punctuation marks or gaps will be there in between the codon, and the sequence is read continuously as groups of 3. So here it is in the form of group of 3, like, so it should be read in the group of 3. For example, AUG, UUU, UCU, yes, like that it goes on. And there are no commas or spaces between the codons, and there are no comma or spaces between the codons. And next one is nearly universal. So the genetic code is nearly universal. This means that the same codon generally specifies the same amino acid in different organisms. For example, if you take this triple UUU, yes, it codes for phenylalanine, and this generally is true from bacteria to humans. So everywhere it is the phenylalanine only. And exceptions are even found in some mitochondrial codons and also in some protozoans. So we say that the genetic code is nearly universal. And next one is the AUG. So AUG has dual functions. So the codon AUG has 2 functions, right? So first it codes for the amino acid methionine. Yes, it codes for the amino acid methionine. Second, it acts as the initiator codon when during translation. So it acts as a initiator codon during translation. So AUG is nothing but methionine plus initiation. Okay. So next one is the stop codons. So the three stop codons. So already we saw what are the three stop codons: UAA, UAG, and UGA. So they do not code for any amino acids. Instead, what they will do, they signal the termination of translation. So they will signal the termination of translation. So now let's see about mutation. Mutation and the genetic code. So what is a mutation? A mutation is a change in the genetic material. So what is it? It's nothing but the effects of, it can be like large deletions or rearrangements. So they are relatively easy to understand. And what they will do, they may cause some loss or gain of a gene, and or they may affect a particular function also, or even a small change in DNA, it can sometimes produce a major effect. And what is this point mutation? Say point mutation involves a change in a single base pair. So we have already seen all these things is in our previous videos. So point mutation. So what happens in point mutation? It involves a change in a single base pair. And one best example for this one is a sickle cell anemia. Sickle cell anemia. So what happens? So in the gene coding for the beta globin chain. So beta globin chain, a single base pair change causes a change in the amino acids. So we already saw that how that glutamic acid GLU, it is changed into valine VAL. So this change in the beta globin protein results in sickle cell anemia, right? So next we are going to see the frame shift mutation. So let us understand the effect of insertion and deletion of bases. So let's see these statements. Yes. So the reading frame analogy. So this is the original statement. RAM, yeah, HAS, RED cap. This has some, this sentence, yeah, it is like a sentence, right? Ram has red cap, right? It's nice. So let's see the next one. So what they have inserted. So here, if we insert one B has been inserted here. So where they have inserted the B here. Yes. So what happens? You can see how we will read by 3 codons, right? So RAM has, but as the B is inserted here, BRE, DCA, yes, it goes on. And what happens? The reading frame changes from the point of insertion. So it is changing right from the point of insertion. If we insert two letters, a BI, so again what happens? The reading frame changes. You can able to see here. But if we insert three letters, so the reading frames remain unchanged. Yes, it is remaining unchanged. You can see Ram has big red cap. So insertion or deletion of one or two bases, frame shifts. You can see here frame shifts. Yeah, insertion of one or two bases, frame shifts. Insertion or deletion of three or multiples of three bases. So the reading frame remains unchanged. So this is a very easy rule now. So next we are going to see tRNA, the adapter molecule. So now we come to an important molecule involved in the translation, that is the tRNA. Otherwise, it is called as the transfer RNA, right? So Francis Crick, he realized that there must be a molecule that would perform two functions. What are those? First one is the read the genetic code, and also link it and link the code to the correct amino acid. Yes, first one read the genetic code, then link the code to the correct amino acid. And this molecule is called as an adapter molecule. And that adapter molecule is nothing but tRNA. So think of a tRNA as a translator and delivery vehicle. Yes, the mRNA contains the instruction in the form of a codon. And the tRNA recognizes the codon and brings the correct amino acids. Let's see the structure of tRNA. So the tRNA molecule has two important regions. One is the anticodon. This is the anticodon, right? So this anticodon loop contains the anticodon. And the anticodon is complementary to the codon is complementary to the codon which are present on mRNA. Yes, this is this. Okay. And then what happens? See the codon and anticodon, how they are pairing. Yes, they pair through the complementary base pair. See AU, GC, UA. Yes. So next one is the amino acid acceptor end. Where it is? This is the amino acid acceptor end. So the other end of tRNA, we call this as the amino acid acceptor end. And this specific amino acid attaches to this end. So this is a specific amino acid which is attaching here, and this is another specific amino acid. So now how it is? So this is the amino acid, and this is the amino acid acceptor end, and this is tRNA, and this is the anticodon loop, and this one is the reads the mRNA codon. Yes. So this tRNA, they are specific for particular amino acids. So this means that each tRNA is associated with a specific amino acid. What it means? This means that each tRNA is associated with a specific amino acid. So for initiation of a translation, there is a special initiator tRNA. What it does? It recognizes the AUG initiator codon and helps begin translation. And there is no tRNA for stop codons. Say there are no tRNA for stop codons because the stop codons, what are the stop codons? We already saw in our checkerboard, right? So it is a UAA, UAG and UGA. So they do not code for any amino acid here, maybe at the end. So therefore no tRNA carries an amino acid for these codon. So instead they signal the termination of translation. And what is the shape of this tRNA? What is the shape of this tRNA? See, the secondary structure of tRNA, it represents a clover leaf structure. But its actual three-dimensional structure is like an inverted L-shaped structure. Inverted L-shaped structure. Yes. So, exam point of view, what you should remember is tRNA is an adapter molecule, and the anticodon loop, it recognizes the mRNA codon, and the acceptor end carries specific amino acid, and the initiator tRNA helps start the translation process, and no tRNA for the stop codons. And also the secondary structure is a clover leaf, and the actual structure is inverted L. So we have come to the conclusion part. So finally, let us revise this. So whatever we saw in the slides. So we know about the codons. Yeah, triplet code we know, and what is a degenerate code, and also universal code. So from bacteria till the human beings, but still some few exceptions are there. And also AUG is the start codon. Yeah, it is the initiator codon, and also codes methionine, and also stop codon. These three are the stop codons. And point mutation is a one base change, and the frame sheet is, it can be an insertion or a deletion of non-multiple of three shifts frame. And the tRNA is an adapter, so it reads the codons, carries the amino acid, and anticodon, they are tRNA triplet complementary to the codon. Yes, we have come to the conclusion part. Don't skip the video, watch the full video. We'll see you in the next video. Thank you.